You see the difference of height, eh? Our previous professor did not use the kürsü neydi? Podium. I will have to use a podium. I will not be talking about genetic directly, but I believe that my topic will bring questions into the minds of my friends who are interested with genetics. I would like to talk about quantitative EEG. Psychiatric drugs and treatment resistance are very closely related. There's a 30% treatment refractory patient percent. This is a very important problem for many drugs. Plasma levels of drugs, of course, in terms of pharmacodynamics, is very important. For the drug to reach the brain and show its effect, there is a pharmacodynamic process, but sometimes we also have the difficulty of relating plasma levels with its effect. But sometimes we can understand the effect of the drug by seeing the drug's effect on the brain. What was our goal? Quantitative EEG, pharmacoEEG, could be used in differential diagnosis as a laboratory instrument. Our second goal was as follows. We are treating people and there is a genetic question mark here. Are we producing a stable effect? There are many electrophysiological markers which can be detected with quantitative EEG. I want to first go through the data obtained from healthy control. This is a study we conducted at Cerrahpaşa University. Schizophrenia, dementia patients were studied. These were patients referred to our university between the years of 2005 and 2007. 9 PTSD patients, 14 anxiety patients, 16 panic disorder, 18 major depressive disorder, and as follows. You know the inclusion criteria. What about the methods? 19-channel EEG was used. It is a medieval instrument, but it is still applicable, still very useful. We have published a lot using that instrument. 50 epochs with eyes closed, 50 epochs with eyes open, eyes closed and eyes open. Fast Fourier transformation was performed and percent power analysis was conducted. 76 variables were analyzed. You know, though, of you who know about the statistics, 76 variables you are dealing with. It's like fishing. You are sure to fish something out. Might be the idea that some of you are thinking. I want to go through our study design. There was a baseline, there was a single dose drug, and 1 hour later and 3 hours later responses were noted, after which first week, first month EEGs were recorded. And response criteria were the same for the other treatment protocols. We had controls, healthy controls, and we tried to compare the two. What were the drugs used? For PTSD, we will use sertraline. For diffuse anxiety disorder, milnacipran. For panic disorder, imipramine. For major depressive disorder, amitriptyline. For schizophrenia, we use haloperidol. These drugs are gold standards for their matching disorders. Baseline data. We have a discriminant analysis for EEG. 84.6% of the population was correctly classified, shown by the discriminant disorder. Without any drugs, 84%, by 84%, I can discriminate the population without giving any drugs. 1 hour after drug administration, this discrimination power goes up by 95.7%. 3 hours after drug administration, again, very similar to 1 hour result. 1 week after drug administration, discrimination power is 100%. I make no mistake. Zeros are controls, ones are PTSD, etc., etc. When you look at these, no one goes anywhere wrong. Discrimination power is very sexy. 4th week results, 100% discrimination power. What is this discrimination? You see here, shown in different colors, you see the disorders. Between 4 and -6 and -3 and 4, there are 2 axes. Yellows shown as 5 show us schizophrenia. Schizophrenia patients seem to have a different domain, and the other disease disorders have different domains. Do not forget the axes, because these axes will change in a while. Again, everybody is being discriminated, but the axes are changing. That is, the formula is changing. What does the formula change to make us think? When we use quantitative EEG, can we diagnose a patient? No, we cannot use it for diagnosis, but everyone goes to their correct spaces. However, the numbers on the axes change. We have to make a correction for the diagnosis. We are currently working on this correction. First week data, first month data. The groups are more clear. Every disorder has its own space, its own cluster. The results. QEEG had discrimination power at every stage. This quality was more prominent for the first week and the first month. EEG data used with the psychiatric diseases is not a state-dependent marker, it's a trait-dependent marker. It is a genetic profile. It shows us something about the genetic profile. It shows us how the brain of a person works. If a person's brain works in a specific way, then the person's brain works in that specific way in the continuum of the time. It never corrects. I think it was short.